Hey everyone, and welcome back to the second part of this video on 3.1 of Higher Biology. That's Unit 3, Sustainability and Interdependence, and Key Area 1, which is Food Supply, Plant Growth and Productivity. So as I mentioned in the previous video, I've split this key area up into two parts because otherwise it'd be really long and you'd be switching off halfway through it. So in the last one, which was 3.1a, we discussed food supply. We had a think about the impact of growing human population and how we're going to produce enough food for them, but also how we're going to produce food in the most sustainable way. So if you've not watched 3.1a, you're on the wrong one. Please go back and watch that one first. And then this is 3.1b, which is going to be on photosynthesis. So hopefully you remember in the first video, we discussed how everything comes back to plant growth. The crops we grow rely on photosynthesis to grow, but then things like livestock also eat vegetation which need photosynthesis. And even if you are only eating livestock, they ate a producer at some stage and looked at the impact of trophic levels. What we're going to look at today, though, is moving to photosynthesis, and we're going to be having a look, first of all, at how light is taken in by a leaf. So, we've discussed photosynthesis before in National Applied Biology. A lot of this system is going to be very similar. We're obviously just going to go into a lot more detail in higher. So first of all, photosynthesis. It's the process by which green plants trap light energy using photosynthetic pigments. And we're going to be looking at what these pigments are. And they use that energy to produce carbohydrates. So previously we looked at how light energy is taken in by a plant and through a series of reactions converted into glucose or sugar, which was the end point. We're going to start off by looking a little bit more into how that light energy is taken in before we actually go on to the process of photosynthesis itself. So we're going into a little bit of physics territory here, but to start off with, we're going to be talking about the visible spectrum of light. So visible light is the only part of the electromagnetic spectrum which is visible to us. And this visible light can be seen in the form of different colours. And these colours all have a different wavelength. So like you can see on the diagram on the right hand side, the highest or the longest wavelength of 700 nanometers is red, and it starts breaking its way down through all these colours of the rainbow down to the end where we have our blues and such at 400. And this is important because we're going to be seeing how this visible light is taken in by a plant. So just to recap, if people aren't familiar with visible light, if you have this idea of light striking a prism, it separates out into all these visible colours that we can see, which again are all different wavelengths. We start off with red, orange, then we have yellow, green, blue, indigo, and violet. You might remember going back to maybe first year science, Roy G. Biv and the colours of the rainbow that go on. So the highest wavelength is red, and as we go down to our blues or indigos and violets, it gets smaller, it decreases in terms of the wavelength. Now don't worry, we're going to be applying this to plants in just a second here by seeing how this light is actually taken in by different pigments. So first of all, in terms of a leaf and a leaf taking in any of this light, we have all this sunlight hitting a leaf, but not all of it is going to be absorbed. So what happens here is that 83% of the light, so the vast majority of the light, is absorbed by the plant, and we're going to be using that for photosynthesis in a minute. But there's also two other things that can happen. So it can be absorbed, but the light can also be reflected, so bouncing off it, or it can be transmitted, which is where the light goes through the leaf. Now, as you can see in the diagram, 12% of it is reflected, 5% is transmitted, but you need to know what can happen to light that hits a leaf. It's not all observed, it's absorbed, it's either absorbed, reflected, or transmitted. And the easy way to remember this is ART or ART. However, in terms of talking now about pigments, about that light that has been absorbed, what we're going to think about here is what is a pigment. And it's maybe something you've heard before, maybe about your skin or colorations, but obviously we're focusing today on plants. So a pigment is any substance that absorbs that visible light that we've been talking about. Now, green plants contain a few different photosynthetic pigments, and what that does is it enables them to absorb different wavelengths of light, which is what we're going to focus on just in a moment. However, most leaves actually reflect green light. A lot of the green light that hits a plant is reflected off instead of being absorbed, and because of that we perceive them to be green. So quite an easy way to remember what's the part of light that the least is taken in by a leaf. You think of a leaf as green because that light is being bounced off, it's being reflected. Now, we're going to talk just here about different types of these pigments that we've been discussing. And the first couple of ones you should recognise the name of. So, as I said, green plants contain different photosynthetic pigments, and these absorb light energy for photosynthesis. That's the function of them, that's the job of them. There's chlorophyll A and chlorophyll B, so both of which are just different forms of chlorophyll, and they are our major pigments. They're the main ones that absorb the vast majority of light. They're really, really important. But there's another two, and we call them the carotenoids. Now, the carotenoids are made up of two pigments called xanthophyll and carotene, and we call the carotenoids accessory pigments because they're almost an add-on to the major pigments of chlorophyll A and B. So first of all, just try and get into your head that chlorophyll A and B are the major ones, they take in the most light. The carotenoids, xanthophyll and carotene, they are accessories, they are added on. But each of these pigments absorbs a different range of those wavelengths of light that we looked at. And what this does is it allows photosynthesis to take place over a wider range of wavelengths. So to put this into practice here, if we look at chlorophyll A and B, if we look at the light that they absorb, and on the scale of absorbance here, what we can see is the majority of the blue and the sort of indigos that we looked at the left-hand side, that's taken in by chlorophyll A and B. Then there's a big gap where there's not much absorbance going on at all until you're hitting the sort of orangey and mostly the reds. So mostly blue and red wavelengths are taken in by the chlorophylls, by chlorophyll A and B. And if you're looking at the colours at the bottom, you can see that green light. There's not a lot going on there at all. It's not being taken in by the chlorophylls, and that green light is being reflected or transmitted by the leaf, giving that green colour that we discussed. However, these accessory pigments, the carotenoids that we're talking about, they take in another range of colours, some of which include green. So if you look now at this diagram at the bottom, you can see the carotenoid wavelength has also been added in, and that goes past where chlorophyll B effectively stopped absorbing and goes into this green area. It doesn't take in tons of green, it doesn't take in the full range of green, but it does take in some green light. So the point of using these carotenoids, I'm going to repeat this a few times, is that it extends the range of wavelengths absorbed. So it makes more of the wavelengths available to be used and more light being absorbed by the plant. And they pass that energy onto the chlorophyll for photosynthesis. So before we go any further, we're going to look at two different types of spectrum, or the two of them are spectra. Now you can come across either of these and you need to know what they actually mean and what the difference of them is. So the one on the left here is the one we've just been talking about. It shows you the absorption of those wavelengths of light by the different photosynthetic pigments. Now, this is called the absorption spectrum. So it's a spectrum showing you the range of absorption. So that should be fairly straightforward for you to remember. And all it does is show the absorption of each photosynthetic pigment at different wavelengths of light, like we've just been studying, comparing the reds and the blues and the greens. The second spectrum we're going to look at, though, if you look at the axis, talks about the rate of photosynthesis. So this is the rate of photosynthesis at each of those wavelengths, and we call that an action spectrum. So this shows the rate of photosynthesis at each wavelength of light by the plant. And again, you can see there's the vast majority of photosynthesis is taking place in the blue and in the red areas, and there's a major dip in the middle. So again, when comparing these, you can see that both of the spectra are similar because they peak in the red and blue areas, where the chlorophyll A and B absorb the majority of light. So that's great, we can see that, that makes sense. However, what we can see is that some photosynthesis is taking place in the green and yellow wavelength. It's not that there's no photosynthesis taking place whatsoever. That's because those carotenoids absorb some of those parts of visible light that chlorophyll does not, and it passes that energy onto the chlorophyll during photosynthesis. And again, the point of using those carotenoids is to extend the range of wavelengths that a plant can use for photosynthesis. So that's really important. Those accessory pigments, those carotenoids, the xanthophyll and carotene, are there to extend the range of wavelengths used. Okay, now we've looked at how light is taken in the different pigments, it's really important. We're going to take a closer look at the structure of a chloroplast. So this is a bit like when we looked at Unit 2 and we looked at cellular respiration and we were talking about the mitochondria. And we talked about how the mitochondria have a double membrane, and we looked at the electron transport chain and the different parts that were used for the different stages of respiration. Back then we discussed that chloroplasts have a double membrane as well because of the use of enzymes and such in important reactions. So this is the time we're going to talk about this in photosynthesis. So chloroplasts, as you can see from the diagram here, have both an outer and an inner membrane. Now there's a couple of parts that you need to know here because it tells you about which part of photosynthesis is taking place in each area. So first of all, there is the granum, or the grana, if there's many of them, and that's the site of stage 1, which is the light-dependent stage, or light reactions. And that contains chlorophyll, because we all know that chlorophyll is there to take in light energy. So that's going to become really important, and we're going to talk about stage 1 in the granum first. Second of all, there's an area outside it, which is called the stroma. And the stroma is the kind of darker area. It's not one of these little stacked green discs. It's the site of carbon fixation, which hopefully from National 5 you remember is stage 2 of photosynthesis. And that does not contain chlorophyll because it does not require any light. What the stroma does use is products from the first light dependent stage and also carbon dioxide from the atmosphere. It doesn't need to take any light at all. And there's also little almost bridges between all the grana, which is called the lamellae. And these are used to just link the grana together. And again, they're structural. They do not need any chlorophyll whatsoever. So first of all, make sure you know these parts, but I'm going to mention them again as we talk about the different stages. Okay, so when you come back to this is probably going to be a bit more useful as a bit of a summary of what happens in the two stages of photosynthesis. For now, part of it, especially carbon fixation, will just look a bit alien to you, and that's totally fine. So in terms of the stages of photosynthesis, this is very similar to National 5. We've got the light reactions as stage 1, and we have carbon fixation as stage 2. Now, in the first stage, light energy from the sun is absorbed, but we know it's now absorbed by those photosynthetic pigments we've just been talking about. And this energy is going to be used to generate ATP, which is going to get moved on to stage 2, but it's also going to be used to split water into hydrogen and oxygen. And what you might remember is that hydrogen is passed on to stage 2 and oxygen is released from the plant and goes into the atmosphere, which we breathe. So again, from National 5, that should be fairly similar. We obviously go into a little bit more detail. Now stage 2, which we'll cover afterwards, is very different. Again, it is that cycle of enzyme-controlled reactions that we talked about in National 5, but again, there's a lot more detail here. We have an enzyme called rubisco, which is used to fix carbon dioxide to a molecule called RUBP to produce 3PG. This 3PG is phosphorylated by the ATP that we've produced from the first stage, and then it's combined with hydrogen, again from the first stage, in order to form a molecule called G3P, which is then used to either synthesize glucose or regenerate RUBP from the start. Now, again, as I said, the first time you come across this, you might think, what does that mean? Don't worry, we're going to go through these one stage at a time. Okay, so first of all, in the light reactions, absorbed light energy is used to excite electrons in the pigment molecule. So we use this term quite a bit in terms of excitement. These electrons are now very active, they're excited, they're basically bouncing around like a hyper child. And just now, what's happening is that the transfer of this electron through the electron transport chain releases energy to generate ATP by the molecule or the enzyme ATP synthase, which you might remember, or hopefully remember, from our Unit 2, where we covered cellular respiration. Okay, so let's now move on to the light reactions and the process of this. So we've just been talking about light energy being taken in by the chlorophyll and creating these high energy electrons. So what now happens, and this diagram is trying to show these electrons becoming excited, are passed on to this process here, which hopefully you remember the structure of the electron transport chain. So these high energy electrons are passed on to the primary electron acceptor, which is basically the start of this process. It's the first part where these electrons are taken in or are accepted. So hopefully that's useful for you trying to remember the names of this. What then happens is the electrons are passed along this transport chain and pumping hydrogen ions into the grana. Now, as it works its way across, again, hopefully remember even the structure from the previous lesson, we have this ATP synthase enzyme, this really important enzyme that is there to synthesize ATP. As those hydrogen ions are moved along, they're used by the ATP synthase in order to generate or to synthesize ATP. Now this ATP, this energy, is going to be passed on to stage 2, carbon fixation, where it's very important there. However, some of that energy is also going to be used in order to split water through a process called photolysis. So if you think about it, every plant needs water. If they don't get water, they're going to die. We covered that a little bit in the transport systems in water PowerPoint from National 5. Now, one of the reasons why plants need water is for photosynthesis. Now, this is where it is needed and where it is required in photosynthesis. The water, H2O, is split up into hydrogen and into oxygen. And as we mentioned earlier, that oxygen is a byproduct of this reaction. Plants do not need it, so it's just passed out of the leaf and it diffuses into the atmosphere. However, hydrogen is needed, and we need that for part 2, for carbon fixation. Now, the other parallel we're going to draw with the respiration stage of higher biology is that hydrogen does not just go into stage 2 itself. It does not exist as its own molecule, which is then used through the process. Instead, it is carried by a coenzyme, and a coenzyme called NADP or NADP. So you might remember that previously in respiration we talked about NAD. You had NAD, which then was NADH as it transported a hydrogen. We have a very similar coenzyme in photosynthesis, but it's called NADP. And similarly, when it transports a hydrogen, when it carries that hydrogen across into the next part, it becomes NADPH. Now, I try to remember the differences of them, but instead of NAD, by NADP, P for plant, or P for photosynthesis, and that's the difference between them. But regardless, that NADPH then carries the hydrogen into carbon fixation, and that ATP that was produced by the ATP synthase enzyme now gets passed to carbon fixation as well. I just went into a little bit more detail here just to talk about that water being split. So photolysis, just in case you come across the term, photolysis just meaning breaking of water using light, is the water is broken into oxygen, as we said, releases a byproduct, and that hydrogen. And it's just key to get into your head that the hydrogen doesn't transport itself, it's carried by the NADP coenzyme in the form of NADPH onto the next stage. This is meant just as a little summary for you so far if you're working your way through this, and obviously this is the first stage, light reactions, just to see exactly what's going on. So far, light energy has been absorbed by a pigment molecule. The electrons in that pigment molecule become excited, and the excited electrons are captured by that primary electron acceptor of a chloroplast, which starts off this whole process of the electrons being transferred through an electron transport chain, and the released energy is used to generate ATP through ATP synthase and used to split water into oxygen and hydrogen through the process of photolysis. So this is quite a lot to take in, so I hope you're pausing this video as you go along, or maybe you're just using this to revise and run through the process again. But what we're now at in this stage is the end of stage 1, where we effectively have 3 products, but only 2 are going to be used because only 2 are required for carbon fixation. So that oxygen has been released into the atmosphere as a byproduct. That ATP that's been generated through the ATP synthase enzyme has now going to be moved on into carbon fixation, and that hydrogen in the form of NADPH is also going to be used. Okay, so now we're moving on to the final part of photosynthesis, carbon fixation. So hopefully you remember from the chloroplast diagram earlier on that carbon fixation takes place in the stroma of the chloroplast. And the stroma does not have chlorophyll, it is not light dependent, meaning it does not require any light. However, this process does consist of several enzyme-controlled reactions. So again, going back to National 5, we looked at this cycle of enzyme-controlled reactions which use carbon dioxide and the products of stage 1. Now that's still broadly true, we just go into a bit more detail as we do with most of these things in higher. So let's get started with this. As we go through this, I'm going through each stage separately, but then there is going to be a summary cycle which shows you this process all working together in one carbon fixation cycle. Now in terms of cycle, you may come across carbon fixation called the Calvin cycle, named after the scientist who discovered this. Just so you're aware that the Calvin cycle and carbon fixation is exactly the same thing. We just tend to use carbon fixation now in higher biology. But in older past papers, for example, or textbooks, you'll see the Calvin cycle quite a lot. So there's going to be a few different names thrown at you, which you might remember from when we went through the summaries at the start of photosynthesis. So during this process, we're going to start off with a molecule called ribulose biphosphate. So this ribulose biphosphate, we abbreviate to RUBP. I'm going to tell you the full name once, and then I'm going to go through just the smaller version of it, the abbreviation. So we have this molecule RUBP, and we want to convert it into something called 3-phosphoglycerate, and that's called 3-PG. To do that, we use an enzyme, and this enzyme is called Rubisco. And the role of Rubisco is to fix or bind carbon dioxide to that RUBP. So we have RUBP, and we have the carbon dioxide, which is taken in from the atmosphere, and the enzyme Rubisco binds them together in order to go through a process and result in the formation of an intermediate compound called 3-PG. So again, make sure you go through this a few times just so you get these ideas in your head, and I'll just work through it one piece at a time. So then we have this 3-PG, which is a really important intermediate molecule, and that is going to combine with some of our products from stage 1. So first of all, 3-PG gets a hydrogen, remember from the NADPH. So that comes across, it deposits a hydrogen which attaches and combines to 3-PG. However, we also had that ATP that was produced from the ATP synthase in stage 1. So this 3-PG, which now has a hydrogen as well, is going to become phosphorylated by ATP. And hopefully remember, phosphorylation is where that inorganic phosphate from ATP is provided, resulting then in ADP, adenosine diphosphate, leaving. So the 3-PG has effectively been given a hydrogen, and it has been given a phosphate. And what it's now done is this has created a molecule called G3P. So G3P is what we want to get at basically at the end of this process. So again, 3-PG, adding a hydrogen, adding a phosphate, we now have G3P. Now, this G3P can either be used, and the vast majority of it is used, in order to synthesize glucose. So this is what we wanted to result at the end of photosynthesis. You have now produced glucose, which the plant can use for energy or for different things, but we'll look at in a couple of slides. However, because this is a cycle, some of that G3P can be phosphorylated again, and as it does so, it is used to regenerate RUBP from the start, which hopefully remember we can call it a carbon dioxide acceptor because it bound with the carbon dioxide through Rubisco enzymes. So this then regenerates the amount of RUBP that can then continue the carbon fixation cycle. So again, that G3P is going to be used in order to make glucose, which we want, but it can also be used to regenerate RUBP. Now, it might be worth you going through those slides again, just to get those ideas in your head and these different names, because they sound very similar and you need to know the differences between all of them. However, this diagram here, I feel, is a good way of summarising what happens, and hopefully you'll be able to do this from your own memory fairly soon. So again, just a brief run through here. If we look at the bottom, we have this RUBP molecule. It then binds with carbon dioxide through the use of the enzyme Rubisco in order to make an intermediate compound called 3PG. With 3PG, it is phosphorylated from the ATP, and it also receives a hydrogen from the NADPH, which then leaves as NADP. After that coenzyme has left, after the phosphorylation has taken place, we now have G3P. And G3P, the majority of it is going to be synthesized into glucose, but some of it is going to be phosphorylated again in order to regenerate RUBP. And this cycle keeps going on, as you can see, just continuously. Now, finally, we're going to look at glucose. And you might remember from National 5, we talked about sugar and what it can be used for. We're going to effectively say the same thing here, but there's just a new one for you. So finally, this last point of this massive key area, what actually happens to that glucose. So some of that glucose that's made through this whole process of photosynthesis that we've just studied, that can be used in respiration in order to provide energy. Now, if it's used to provide energy, some of it can then be synthesized into complex carbohydrates. So the main thing, let's focus on the main point here, is that used in respiration to provide energy. So you've covered respiration already, you know that glucose is required for respiration to take place, and that's a whole other stage that you can go back and revise now. So the glucose can be used as a substrate for respiration, or it can be converted into the storage carbohydrate starch. So remember, the form of storage in plants is they don't have fat to store excess energy, they have starch instead, so it can be stored for later on. It can also be used for structure. So if you remember going back to plant cell walls are made up of cellulose. And if it's made up of cellulose, that is a structural carbohydrate that's been derived from glucose. We've spoken about those three at National 5 as well, but there's one more we're going to discuss, which is quite a broad part, and it can be used for biosynthesis pathways. Now, what happens here is that glucose can be formed into metabolites such as DNA, amino acids, proteins, or fats. So that's a huge bulk of things that can be made up, but very much building blocks of life that need to be made. And again, the plant has created, it's generated this glucose, and it's going to use them for all four of these different parts that you need to be aware of. And that is us for this key area. I know it's been absolutely huge. Thank you very much for listening through it. Hopefully it's been useful for you. You might need to go back, especially through the stages of photosynthesis, to try and consolidate your notes. But we covered the food supply part in the 1A video. This is now on 3.1b. So just make sure you're aware of the photosynthetic pigments, what they are, what they do, what is the point of having carotenoids. Then have a look at the comparison between the absorption spectra and the action spectra. Have a look at the different parts of the chloroplast, what happens in stage 1 light reactions, what happens in carbon fixation, and then what can you use that glucose for. Thank you very much for listening through this, folks. I really appreciate it. And thank you again for all your comments that have been coming in. Hope this is going well for you, and we'll continue on with the rest of this final unit of higher. Thank you very much for listening.